gene for the same trait. Now, in this duplicated chromosome, how do I call it? One of them, of course, is a chromatid. Now that still joined together, we're going to see this is a centromere. We refer to these guys as the sister chromatids. In the same way there, we refer to these ones as the sister chromatids. So then, the relationship between this chromatid here and this one here, I refer to them as non-sister chromatids. They're not sisters, no. I don't know why they don't say brothers, but they're referred to as sister chromatids and non-sister chromatids. Let me see if the next example can help. Okay, let's look at it. Yes.
My network was tripping. What did you say the homologous chromosome is?
Okay, so now it means, do you know that you have to go back to this? It was tripping from this end.
No, just only the definition part.
No, but then I explained it on this slide. So what we are calling a homologous chromosome, because this understanding must be on this slide. Anywhere else, I was trying to explain the relationship between sister chromatids and non-sister chromatids. But anyway, so we're trying to say the chromosomes in an individual like yourself and me, they exist, in pairs. And we said this pairing is not just at random. It's based on the fact that the chromosomes that pair, they carry the same combination of genes. Now, remember, I'm saying they carry the same combination of genes in relation to them specifying a trait. For example, if there's a gene R here and a gene R there, if it is in a plant, this is a gene for color red, meaning that on both chromosomes, the one that came from the father and the one that came from the mother, at the same position, there must be the gene for color, meaning that this one contributes to the color red, this one also contributes to the color red and not anything else. So this is the locus for color. When you go to this position, maybe this is for plant height. Okay, if it is in beans, maybe the pod should be rough or smooth. So meaning that the trait that this gene should contribute to is the size of the pod. This trait from the mother must also be the size for the pod. In short, these are homologous chromosomes based on the fact that they carry the same combination of genes, or those combinations of genes specify the same trait. One other thing you're going to note is about homologous chromosomes, they've got the same height. And also we're going to look at a feature referred to as a centromere. The centromeres of those homologous chromosomes will also be at the same position, okay? So the location of genes on homologous chromosomes is the same, and those genes carry or contribute to a similar trait. And the trait is a visible feature that you're able to see or measure. For example, right now, you can go and check your blood group, and that's a trait because it is something that we can measure, but also we can trace it back to information on your chromosome. In fact, if you're blood group O, it means that, some of your parents must also be blood group O. For example, for me, I'm blood group O. My husband is blood group O. All our children in this house are blood group O. But it means that both of us on the chromosomes that specify blood group have paired, and that's how come we've got children that have blood group O and things like that. So I hope that is clear. Now on this slide, what I was trying to tell you is that if these two are homologous chromosomes, we understand what that means. Now, remember, these chromosomes, they duplicate. That is how come we can heal, we can grow, and things like that. So in this chromosome from the father duplicate itself, it has got another pair. This guy, the partner, is called a sister. They're now referred to as sister chromatids. They don't refer to them as brother chromatids. I don't understand why, but they're referred to as sister chromatids. So meaning that sister chromatids carry the exact information that was on the parent chromosome before it duplicated. These ones came from the mother. When it duplicates, it makes a copy of itself. It also referred to as sister chromatids. So sister chromatids carry the same combination of genes, and also they are held at this position like an umbilical cord that is referred to as a centromere. So sister chromatids will always be held at a centromere. But one thing, they're going to have the same height and also same combination of genes. Now, the relationship between this chromosome that has duplicated itself and this chromosome, if I look at these arms of the chromosomes, these are non-sister chromatids. Okay, these are sisters together, but if you look at this chromatid and this chromatid here, these are non-sister chromatids. Okay, non-sister chromatids, but together within themselves, these are called sister chromatids. I wanted to use the next slide to see if I can, on this one, talking about non-homologous chromosomes. Now, if we have understood what homologous chromosomes are, then explaining what non-homologous chromosomes should be easy. For example, you have got this chromosome here. At this position, it is carrying a gene for, let's say, height. This other chromosome, they kind of seem to have the same height, but it's carrying a gene E. Okay, maybe this one could be for, if this one is for height, this is for color. These two cannot pair. Why? Because at this position, they're carrying different genes or genes that specify different traits. So when you've got such chromosomes, refer to them as non-homologous chromosomes because they're carrying genes for different traits at different locations. And non-homologous chromosomes, this is a chromosomes that are not carrying genes for the same trait. An example, A could mean the height of a plant. And this position here at that same locus, maybe this one is carrying a gene for pod color. Color and height, these are two different traits. And on both chromosomes, they're sitting on different positions. So these are referred to as non-homologous chromosomes. When these duplicate, these two are going to be referred to as, when this chromosome duplicates, this is what we're normally seeing in our slides. This partner here is a sister chromatid. Sister because it came from this guy, okay? And you can see at this locus, we say this is loci. This is the exact address of this gene. This is eye color, height color, or plant height. It's the same thing, isn't it? Because of that, these are referred to as sister chromatids. This also, at this position, this could be maybe pod size or the shape of the seed. If this is for the shape of the seed and the same shape of the seed, and because it came from this chromosome, these two are referred to as sister chromatids. But if I get this chromatid and this chromatid, these are non-sister chromatid. These are non-homologous chromosomes. Why? Because at those loci, they carry different genes. These can only be homologous chromosomes if we have AA, and when you go this side, you also find the same gene for the trait. It should also be AA or maybe small letter a, it doesn't really matter, but as long as at this same position, they should carry the same genes for the same trait. But in this case, we have got non-homologous. Non-homologous because when you compare the locations of these genes, the combinations of genes, they are different. They specify different traits. So this is just like a summary of comparing homologous chromosomes from non-homologous chromosomes. Here, if we say these guys are homologous chromosomes, the size, the height will be the same. What you see here is a centromere. So if there's a gene A, capital letter A, there must be capital letter A there. If there's a gene small letter a here, there must be a small letter a there. Now, these can only be sister chromatids if they carry the same combination. That is to say, if there's a small letter here, there must be a small letter there. If there's a capital letter here, there must be a capital letter there. That is to say, if it is dominant, must be dominant. Recessive must be recessive. That's what I'm trying to say. So these are homologous chromosomes. Why? Because they have got the same height and the same combination of genes. These two also, they are homologous chromosomes, okay? Because maybe they have got the same height and the same combination. But the relationship between this chromosome and that chromosome is non-homologous because they have got different combination of genes. In fact, you can even see the centromere position there is very different from the centromere position there. So meaning the genes that are found on this chromosome must be different from those genes there. So here you're learning about homologous chromosome, what a non-homologous chromosome is, and also to understand that this is a chromatid, this is also a chromatid. These two are sister chromatids. This is a chromatid, this is a chromatid. Because both of them are together held by the centromere, these are sister chromatids. But this chromatid and this chromatid, these will be non-sister chromatids, okay, in a similar way. This is a sister chromatid to this one, sister chromatid to this one. But these two here are non-sister chromatids. Despite them being homologous chromosomes, they are non-sister chromatids. Chromatids are held together at the centromere. Okay, sister chromatids are held together at the centromere. Non-sister chromatids, they don't share a centromere. Okay, the centromere is this position here. They're like an umbilical cord of the chromosome that has duplicated. When you think of a sister chromatid, they must share a centromere. So for example, these are sister chromatids. Why they share a centromere? These are sister chromatids. These two are non-sister chromatids because they don't share centromere. That's the easiest way of me explaining this. Okay. So what is on the chromosomes? We know they're genes, right? But now we want to extend this understanding to describe alleles. We want to find out what is a gene and what are alleles. What are genes? We know genes are on these chromosomes because from way back we're looking at DNA carrying genes and things like that. So what is on these chromosomes? We want to distinguish genes from alleles. Okay, at this
Excuse
point
me,
we
but
know. Yes.
sorry to bring you back. I just have a question on the sister chromatids. According to
Go
what you
ahead.
have explained, the sister chromatids, they have what we call the centromere. So, what's the function of the centromere on the sister chromatids?
Okay, so you know some questions that you ask, you can even go and read about them, honestly speaking. Even if I didn't tell you the function of centromere, that's what the question you can ask me to break a lecture. No. Honestly speaking, no, you're being lazy. Okay, but I'll tell you what the function is. So the function of a centromere, first of all, is to hold the sister chromatids. And during mitosis, you're going to see that the centromere is going to be a point where the sister chromatids are going to split so that these chromatids can be allocated to the different cells. The centromere plays important function. Just apart from just holding the sister chromatids together before they can split, it is also a location where the spindle fibers are going to organize themselves. So now, the question you're asking, honestly speaking, is something that you can go and read about on your own. And then if you don't understand, you can come back and say, no, I read about the definition of centromere. No, no. So some things that you're asking me, guys, let's also start, motivating ourselves to read. Understand? I'm not trying to say I cannot answer. I can. The point is that some of the things that you're being given, they're simple definitions that even if I gave my child who's in grade 7, they can give you the answer. So encourage yourself to read. In mitosis, we're going to look, even in meiosis, we're going to look at the centromere and how, the centromere also helps in, ensuring that the two sister chromatids go to opposite ends of the poles. It's actually going to split at some point to allow for cell division to take place. But anyway, I'm hoping that they're going to go back and read. So where was I? Yeah, so I was trying to say,
Madam,
yes.
I was kind of asking if we can stop here because there's a group that is supposed to have chemistry.
Oh, okay. That's fine. Thank you for letting me know. I think we can continue. I just have a few slides. So now, guys, I've shared these notes with everyone. I'm hoping that you can, read through so that even when you come, if you've got some questions, you can tell me where you didn't understand and things like that. Otherwise, thank you very much. Have a good day and see you next week. Bye-bye.